Hi, this is Dr. Ben Morrill. Welcome to episode 96 of Reptile Genetics Weekly. Once again, glad to have you here. And just have to say, first of all, thank you so much for all of the happy comments, kind words, excitement, and then also people ordering tests from all the new things that we announced last week. That was lots of fun sharing that with you all, being on live with you all for a couple hours and talking about that. And yeah, very thankful to have more tests. And we obviously know from the orders we see coming in that that's something you all appreciate. And just wanted to say thanks once again. We're very, very happy and excited about that. And obviously also to be able to offer it at a lower price. Many of you have said you appreciate that as well. And that makes good sense, and that's why we wanted to do it. And it's only possible because you all are ordering from us. That, like we talked about last week, that volume of samples coming in, as that gets larger, that makes it so we can reduce the price. And so just I'm so glad after coming up on 2.5 years, didn't know for sure how it was going to go, and just so thankful that we keep getting more samples coming in, more tests, and more excitement. And so we're very happy, very thankful to be where we're at now. And it's because of you all, and we appreciate it. As far as updates for samples for this week, we have some pretty bad weather on the east, especially southeast or northeast. I mean, we're just kind of getting part of. Actually, I think it is even the southern part. Anyway, East Coast. There's quite a few of us that got dumped on with snow, and I was talking to someone in Texas. They said they're having kind of crazy weather there. It's not snowy, but anyway. I believe we'll have results going out every day this week, and I believe that the panel test, I'll be able to get that run. So we'll have results starting to go out this weekend. But it is possible. I know at least a couple of the shipments I have for reagents and things are delayed because of the weather last night. It'll take an extra day or two to get to me. So there might be something that'll affect us and make something take another day or something. But as of right now, the beginning of the week on Tuesday, it's looking like results will go out every day for fast testing. Sex determination will be a couple times this week. And then the panels, like I said, should have those results starting to go out this weekend. Monday, hopefully, will be about the latest. We'll have all that going. So I think that's all I have for those updates. Kayla, how are you doing?
Hey, Ben, I'm doing great. So, okay, just for the record, y'all might hear Potato being very upset because Jay has a half snow day today and is in the room, but Potato cannot handle when one of us is in the bedroom during the day. Like it's the wrong time for it. So, if you want to come over here and be nice to you, that's fine too. But as far as critters go, actually, I figure I'm going to talk about this before we do business stuff. So in addition to doing social media for Rare Genetics, I also work at a wildlife hospital in our town. And we got a cool patient. So I was working the front desk. We get a call from a lady who is like, "Oh, I found a snake in my basement. I thought it was a worm. And I have it under a cup. It's terrified. Pest control isn't coming for like an hour. I'm so scared." And so I'm like, "Oh, okay, it's fine. I'm our resident snake nerd, so that's totally fine. Send me a picture. I'll get you an ID." And from a picture, even though it was really blurry and from like 3 feet away, I could tell it was an eastern worm snake. Just a little guy, gets like maximum of a foot long. And since it's cold out, and we were about to get some weather soon anyway, as Ben said, which the weather is still coming down here in Roanoke. I was like, "Oh, okay, well, we'll just bring him on in. He's probably lethargic, can't put him out anyway, so he can overwinter with our other reptiles, because we don't release him until May." And so when it comes in, the pest control guy brings us in a Chinese takeout box, the tiniest eastern worm snake I have never seen, actually. It is 3 inches long. That's him in the little Chinese takeout thing. Here's my glove. Small.
You said less than a gram, right?
Yes, he weighs less than a gram and had some stuck shed, so it's a good thing he came in. We can help him out with that. But oh my gosh, so freaking cute. Like mind-blowingly small. I've never seen a snake this little before. Cute as a button. Did not want to bite at all. If he tried to, his mouth is probably too small anyway, but the species is just chill as can be and just wanted to burrow and hide. So little tiny guy is just so cool, and I thought you guys would appreciate how little he is. So yeah. So I'm on cloud 9. This is fantastic for me. And that was my weekend. Uh but yeah, so getting to business stuff. Ben and I are, and possibly also Sean, but I know at least us are going on uh going to onto Colubrid and Colubroid Radio this evening. We're going to record with them, and we're going to talk about the and some of the newest tests that we've rolled out, including corn snakes, our very first colubrid tests, and the first one where we started breaking in the new species for morph testing. So very exciting. Be on the lookout for it. If you haven't followed them already, highly recommend it.
I think we're recording tonight, but this is the one that will go live next week.
I believe so, yes. So be on the lookout for it. And yeah, we're going to be talking about that. Probably going to talk about the worm snake there too. So might have some more cool stuff there. But yeah, so be on the lookout. And oh, that's right. I have to move it on this. There we go. So ball python sheds. We do have an updated list of sheds that we do want samples for. So, Stranger, Disco, Sable, Razor. So we've added some new ones on there as well, like Lucifer. We still need some RK Asphalt. Any of these, if you or someone is working with these, even if it's just a pet and you're willing to grab a little shed sample, these are all helpful for us.
Yes, help us round out some of the complexes and get a few that are ones that we have a few sheds, but not enough to know for sure yet. So
yeah,
you can send those in. We'd greatly appreciate it.
Yes. So if you're sending sheds for research, either for the ball python test that we're looking for, or for new species or morphs in species that we're starting to get into, like corns, be sure to just send us just a little quarter-size sample of it. The species, animal name and name or ID, known morphs, possible morphs. It can be a morph combo as well. It doesn't need to just be a single gene. And any lineage info, who produced it, and anything like that, because, well, when we run into situations like what we have today with the NoCo specials, the lineage info is actually helpful in those cases when we're first starting to figure these out. So we definitely appreciate those of you who are helping out, because we really can't do this without y'all.
And we have, since we asked for them, we've gotten lots of sheds in from all of you, and thank you so much. And just like Kayla lined out there, known morphs, possible morphs, lineage information, y'all are doing a great job of having that on the bags or on a piece of paper or card in the bag with the shed, and that's awesome. We love seeing that.
Yes, for sure. So speaking of which, why do we have, like, why are there so many, for example, specials, axanthics, black pastels? We're starting to find this with genetic testing. So why is that happening exactly? Luckily, a geneticist here. So Ben, you want to talk about it?
Well, it's something that from the very beginning, very, very beginning, way far back, some of these things we figured out, like the axanthics, through breeding. When we bred two different axanthics together and got no axanthics, we kind of realized, then there has to be multiple lines, because they're not compatible. At least some of these aren't compatible with others. And so we kind of knew. And in the case with axanthics, they're actually on different, at least three different that we know now. There's still maybe like Jolliffe, there may be others that are different from those three. We just don't have enough samples yet to know for sure. But anyway, now that we can test for VPI and for GCR and MJ, and then also for TSK, if yours is for sure axanthic and it tests negative for all three of those, then it's something different from those. But anyway, so when they're not compatible, then through breeding we can figure out, okay, these all three look like axanthic ball pythons, but they're axanthic ball pythons with mutations on three completely different chromosomes. And that's what we know now with testing. we know they're actually completely different chromosomes. That's why it makes perfect sense. They're not compatible. But like with specials, now we're learning there's multiple lines of specials. We know from publication from Dr. Hannah Sadell, both albino and ultramel, that there are multiple mutations for albino and ultramel. And then yellow belly was another publication she did. So those are proven ones where we know for sure there are multiple mutations that cause that specific morph. Mm-hmm. And in the case of special, which we're going to talk about a lot today, we'll talk about, whether it looks the same or a little different, like with asphalt and gravel do look very similar, but they were different enough that people kind of kept that name different. And with special, that there's enough of a difference with the NoCo line that's something that was kept kind of separate. And now we know there's a good reason why. Yeah. But there's also some that we think are likely. Either we pretty much know there has to be another mutation, or we're very, very much suspecting so. So we have fire first on that list. So actually, this is kind of a bit of an announcement. We're kind of sneaking into the middle of an episode, but there was some guessing that there's more than one line of fire, and it looks like there is. I'm doing the last bit of validation. So starting most likely next week, as long as you all know that watch the show from a couple of weeks ago, and we talked about this whole process, but I'll be validating fire, a second mutation for fire.
Ooh.
So any of you that have had fire tests and you got a positive result and it makes sense, you don't need to worry about it at all. But if you did a fire test with us since that came out and you got a negative and you're still wondering if that is correct, you can go ahead and send that shed back in with the same information, and like that same order information, and we will run the new fire test for you. There's no additional cost. So since it's something we just learned, it's the same thing we did with ultramel. People that purchased the ultramel test a couple of years ago and only got tested for ultramel A. As soon as we had ultramel B, we let people know, hey, just send in the shed again with the same information, the same order info, and we'll run ultramel B for you for free. So same thing here with fire. If you've done a fire test, and like I said, if you got a positive result and it makes sense, don't worry about it. If you got a negative and you think maybe it really is fire, but our test said negative, send it in again and we will run that second one for you for free. I think we'll have it by the end of next week. We should have that second fire test working, and from then on, every fire test will run both, just like we do with ultramel and yellow belly and stuff like that, and albino. So that will just automatically be done. And then if some people always ask when we say things like this, well, what if I just sent it in, 2 weeks ago? Do you still have it? And can you just pull it and run it? You can go ahead and email Sean at raregeninc@gmail.com and say, hey, I just had a fire test done 2 weeks ago and it was negative. And Sean will be able to tell you if he can pull it. But I can tell you right now, if it's more than 2 months old, because the company has grown, we have so many sheds coming in, anything it's more than 2 months ago, we have to discard because we have so many samples coming in now. So if you sent it in more than 2 months ago, then just know you're definitely going to have to send in another shed. But if it's really recent, you can text or you can email Sean, and he'll be able to tell you if he can pull that and run the second fire test. So we'll probably call it Fire B. So the one we've been testing is Fire A, and then the Fire B test we'll start running for everyone probably late next week.
Yes.
So another one that we are close on is the RK asphalt. And when we did the live, there were several people talking about how that asphalt, especially the supers from that asphalt, are different from the normal asphalt. Mm-hmm. We're close on that one. Acid and confusion definitely test differently. The acids that we run test negative for the best confusion test we have. The confusions we run test negative for the acid test that we have. So it seems very clearly they're two different mutations. And so we're very close on confusion, having a test that we can put out. And then we frequently get, it's not a very high percentage, but there's enough tests that we're running. We get some people with red stripe and black pastel that have questions. They feel like they really do have a red stripe, but it's testing negative, or they really do have a black pastel, but it's testing negative. So that'll be the same thing as with fire and with Ultramel. We are still running the red stripe test and the black pastel tests. With black pastel, we did find Barnhart. We have a test for that. But if you test negative for Barnhart and for normal black pastel, but you're still pretty sure it's a black pastel, we do know there's at least one more black pastel. And it's very prevalent in Canada. And then there's some people in the United States that have some that we've triple checked for sure. They're obviously visual black pastels, but they test negative for the normal black pastel and the Barnhart. So I wanted to put this picture up to be able to just kind of lay out for everybody all the things that we know have multiple mutations, and some we're pretty sure do. And then, like I said, with red stripe and black pastel, we're still working on those. And I don't know if it'll be in a week or a month or 6 months or whatever. Whenever we get that additional, test for red stripe and black pastel, where we can finally test for everything, then the same thing will apply. If you've run a black pastel or a red stripe that was negative and you think it really could be incorrect, we'll fix that for you. So
very cool. All right. So it's been a while since we've done a monthly-ish morph spotlight, and we actually brought a friend with us today, who is very qualified for talking about this particular line of special. So I'm going to bring him on now. This is Kyle Frost of Kyle Frost Reptiles.
Hey, thanks for having me.
Yeah,
thanks for coming on.
All right. So, for those of you who know of special, we have talked about before about just the regular special, or AKA the Baker line special, which is what most people have. But we also have talked about the TCR special line before. And Kyle, your specialty is the NoCo special, right?
That's right.
Yeah.
Yeah.
Okay.
So as we're getting ready to talk about that, I even want you to go back in time even more and just briefly in a minute or two, just give people an idea of what brought you to keeping and breeding reptiles, and then what brought you to ball pythons, and then what specifically that kind of history with why you decided to work with NoCo. And as everyone will see today, you've done quite a bit. You've kept it, just NoCo line, and then you've done a lot of really cool things with it. So if you can kind of start it at the beginning and just kind of walk us through those stories, and I'm sure Kayla and I will have some questions for you too.
Yeah,
sure. It's a lot to remember. I guess it's a familiar trope on this podcast, I'm sure. But I loved reptiles even when I was a kid, and always had reptiles. And I had a leopard gecko at one point when I was young and on my own, and I was going into a reptile shop to get crickets, once a week or so, and they had a help wanted sign on the door, and I'm thinking like, no chance I'm getting this. It's like working at a skate shop or something. Like I'm not gonna get it, but I ended up getting the job, and
nice.
It was a cool shop. It's Amazon Reptile Center in Montclair. It's still in business to this day. And no disrespect, but it didn't pay very well, and I was on my own, and so naturally you kind of get into breeding reptiles as a side gig.
There you go.
And I learned kind of the distinction between it's something you never really think of as a spectator in this hobby, is the difference between a wild caught animal and a captive bred one, and learning as I'm working for this shop that there's a difference, and that the wild caught industry's got its baggage, obviously, and justifiably, and then there's captive bred reptiles kind of command their premium, which they deserve, and they tend to thrive better, and they don't have all the wild caught problems, and so I kind of got it. I looked at ball pythons, which were being imported in the hundreds of thousands every year, is kind of a cool opportunity. They make great pets, They stay small, they're cool. They at the time didn't really come in that many different colors, but there were kind of these new pastel and piebald and some of these new founding morphs were coming out. And it was interesting. It was cool. And so you could kind of like take this cool snake that's small and only lays a few eggs, and you can kind of have a big variety of different things. And so obviously I was attracted to it for a lot of the same reasons everybody was. But I kept a lot of different species of reptiles. I had boa constrictors and as a primary thing. And so when I got kind of bigger into breeding reptiles, I had a lot of boa constrictors, and a lot of there aren't quite as many genes in boa constrictors, although there are a lot. And so it becomes a little bit more important in something with fewer genes to kind of have a nice version of those genes rather than just having as many genes as in one animal as you can. And so as I was breeding ball pythons, I kind of applied that same mindset and always made sure to get the nicest example of whatever gene that I was getting I could. And even, some of my founding normal animals were reduced pattern and banded. And if we have time, we'll get into the whole blade controversy, as I consider it. But I did always kind of be mindful of selective breeding as I'm breeding for genetic traits and combinations. And so if I wanted a fire, I got the lemon back line. It seemed a little bit different. The supers were this pure white kind of thing. If I wanted I'll cut to it, the specials, there were Baker's line was the original line, and Marshall Van Thor at NoCo Reptiles had his own line of special, which made a super that was, in my opinion and in other people's opinion, looked a little bit different than the super Baker line specials. And so it was obvious to me, if I'm going to do any kind of special combo stuff, I'll just get the NoCo stuff. And it was being sold at a premium at the time, and well worth it, for something that you're going to put so many years of work into. And so that's what I used as my special, and I've been working with it ever since, without kind of paying much attention to the Baker line or the TCR line. I had always kind of assumed in the back of my head that it might be a different morph than Baker's special. I didn't really think that I would have an opportunity to prove it until you guys came around and started testing these things. But I sent in sheds, and I was thinking with the way that Mystic and Phantom were proving out to be the same things, and Lesser and Butter were proving out to be the same things. That these things with different names proving out to be the same thing. I was kind of skeptical that this would end up being a different thing, even though I kind of saw the difference for myself. I thought maybe it was just my own optimism about my own projects kind of blinding me a little bit, but I did feel that it had the potential of being a different thing. So exciting that it proved out to be a different morph entirely.
Kind of like that same optimism when you're trying to decide what combos you have when you're slitting eggs.
Right. Yeah. You'd be blind to the likelihood that you're not going to get it, but you're still hopeful.
Yeah.
So as far as if you want me to go into the backstory of the NoCo line special from Marshall, I talked a little bit about it proving out to be a different morph, and I had tried to get a bit more backstory from him on exactly the origins of it, and I didn't quite have enough time to connect with him, but I can kind of fill in the gaps and assume that it is the same story as most of these kind of things. And so back in the day, Marshall had a few Genesis mutations, I guess you would call them, morphs that came out came out of Africa that were assumed to be existing morphs already in America. And so he had a a lesser, he had a tri-stripe, and then he had the special. He may have had a yellow belly too or something like that. I don't recall exactly off the top of my head. But he had these few his own versions of these existing morphs. And he used to vend at the shows all the time out here and had this big display. And so I through a friend of mine that ended up actually purchasing his original animal from him. I got my founding animal. I want to say it was 2013 or something like that, but I'm getting off track. The animal itself probably came from back years ago when the United States would import hundreds of thousands of normal ball pythons from Africa for the pet trade. They would go to pet shop wholesalers in 1,000 lots. And if you had an in with any of these wholesalers, you could go down and they would let you kind of sort through their purchase of 1,000 ball pythons to look for something special, no pun intended. But I got to do that quite a few times. It was a fun opportunity to go through. And usually these, you'd go through and you would just be like so exhausted by the end of it, searching through 1,000 different ball pythons, looking at every single belly and head stamp and side pattern, and telling what's kind of just like unique looking or what might actually be a gene. And it's exhausting. Usually I did it several times and picked up a couple things that never proved out to be genes. Usually when you go and you sort through all these lots, you don't find anything other than normal ball pythons. But it was still really fun. And it's something that, I look back on as kind of a fun thing and something that'll, I guess, will never probably happen ever again. So, it was a cool time and a cool thing to kind of be a part of, a treasure hunt of a bygone era. But, better that they're all captive bred here now, for the most part. Anyway.
Yeah.
But I'm sure that's kind of the foundation of of a lot of Marshall's original morphs. And so, I think it was special, tri-stripe, and lesser, and maybe one other. But it was a special that stood out to me as potentially being something different. And so when I did special, I did that one.
Yeah.
And when you picked it up, what were you thinking you wanted to do with it? Did you want to make supers? Was there certain combos you wanted to go for?
So the supers obviously were the prettiest, coolest thing, but I have there's only a dozen or so morphs that I like. I'm kind of a hater in most aspects. I have these things that I certainly like, and everything else I'm kind of skeptical about. I don't know if I want to incorporate this into my 20-year long project. So special is one of those where I thought it would be a cool highlighter morph for a lot of the orange dream, yellow belly, fire kind of combos that I like, where we're having just adding a little bit of extra spice to something kind of makes it stand out a little bit more. So those are the kind of projects that I always liked in ball pythons. And then on top of that, just kind of selectively breeding for reduced pattern stuff and bold pattern stuff at the same time. And so that's kind of what I've done with that. That's it wasn't necessarily the supers though, but I did figure that I would get around to making supers with it. And the fact that they have a bit more pattern to them than, in my experience, the Baker line super specials. If I can add Enchi and Leopard and all of this to it, then then I could imagine a long-term prospect of having some high multi-gene combinations where you can still tell what everything is and it continues to add to it rather than subtract to it, which is possible with a lot of morphs and a lot of combos.
So in that picture you just had up, Kayla, so that super NoCo there, is that pretty common? They look like that, and do they keep that color into adulthood? So you'll get you'll pull up a couple of pictures, and they'll be kind of different looking, but from the actual same clutch. But they all are dark and bold, and they kind of hatch out looking sort of normal, not too spectacular, fresh out of the egg. And then after their first shed and their first meal, it really kind of enhances. And then, as adults, they'll fade a little bit, and they kind of get a couple flecks on them here, like a banana will too with time, which I think is common of the Baker special as well. That's amazing. This Enchi and then the leopard. That's
Yeah, I want to say
amazing.
that was like 800 grams or so, and looking
Wow.
nice.
Yeah,
The leopard's my favorite so far of the.
That is really nice.
I like that too.
I definitely would have guessed on both of those. You'd have to add more than just leopard or just Enchi. That Enchi,
Yeah.
that's amazing.
Right. Yep, just a straight Enchi super special.
So
Wow.
cool.
Yeah,
And I even just like the base level NoCo adds a really nice, like blush of yellow along the back and between those patterns that I really like.
Yeah, they're highly variable. And this, like, here's an example of kind of an older animal where I hadn't had a chance to refine it too much. Is that they're naturally kind of a pretty looking morph on its own. And then some of my other stuff, you'll see like a blade line kind of things too, where they're a bit more reduced pattern, which some of my stuff's getting a bit more into these days. So I don't know if that'll, add to or subtract in the long run, but it's, a direction that I've been steadfast in so far.
Yeah.
That's awesome.
That's cool. So it's
Nice
a leopard NoCo line special. They all seem to have that head pattern. Again, I don't have a lot of Baker special experience, and so I don't know that the same indicators for what a special is, especially in combos, would be the same thing. But I always look kind of for that head stamp, that kind of, I guess you would call it like a clover-looking head stamp. Their bellies will be a little bit more kind of washed out, a lighter pattern, and kind of a jumbled up pattern.
When I first saw it, I thought it. Oh, sorry. Go ahead.
If you were to ask me to describe what makes a special to somebody that doesn't know what they're looking at, it's really hard to to say that there's much there. It's funny to me that reptile people seem to have an eye for picking out a special, night and day, no question about it. But then there will be certain things that at least I see a difference in certain animals, and certain people just can't be told that there's a difference there. So
yeah,
everyone's got different eyes for this sort of thing.
Mm-hmm.
Yeah.
I mean, it's kind of funny, especially with AI becoming more of a thing, like trying to get AI to see what the human brain sees as a pattern recognizing machine.
Right.
I feel like I see that in its own special way in the reptile community, because the things like you talk to Justin Kobilka at Canova, and he's like looking at things, and like even things we don't know that we're looking at. You just look at something and say, yeah, I know what that is. I can't tell you what it is I'm seeing, but I know it is. And a genetic test will prove it out. So
are you saying reptile keepers are special?
Yes. We all knew that.
It's funny to me that you could consider special incomplete dominant, but a piebald recessive with it almost feels like the special is as visually distinct as a het piebald would be in a lot of instances. Maybe that's not the best example, but a lot of genes like that, where they are, the distinction between incomplete dominant and recessive has a lot to do with how we interpret it. And we do have different eyes for this sort of thing from one person to the next.
Oh, yeah.
For sure.
You were mentioning something about your blade project and how there was something with that.
Yeah, so I've had blade. So Blade was a reduced pattern line of clowns from EBN way back in the day. And I had a few of these animals and a few het animals of these since before it was considered to be an incomplete dominant trait. I was a little caught off guard when that was announced at the time. And, maybe a genetic test will prove it to be so in the future. But I had always, like I said, I had always kind of just picked whatever I could pick the nicest example of a gene. And it was always understood that these reduced pattern clowns came from reduced pattern normals, reduced pattern hets. And I had a few other banded or reduced pattern, just not, maybe line bred is not the right word, because a lot of them were original import animals that just had reduced patterns that reproduced that, but in a way where in subsequent generations it would become diluted, but not in a way where you'd get, half banded and then half busy pattern normals. You would get, if you bred a banded animal to a typical animal, you would get these kind of banded-ish sort of, it would become diluted quickly, but not in a way where it would be divided in a 50-50 incomplete dominant type of way. And so I've had quite a few of these bloodlines over the years. And I had Blade, and I considered it to be the same as those, and I still do consider it to be just a bloodline of reduced pattern normal. I'm in the minority on this, and I know that proving a negative is not quite as straightforward as proving out a new mutation. And so if I were to send in tests for something like that, I suppose I would have to send in samples of what is in line with other people's consideration of what a homozygous blade would be and a heterozygous blade, which I disagree with from the jump. So I don't know. It's a little bit frustrating. And again, it's possible that it is a mutation. I think that even 20 years in, it's a little premature to say that it is this or it is that. In my opinion, it's always been just reduced pattern bloodline. And a great one. I don't think that it diminishes the value at all. In fact, I think that if you want to continue to make nice looking blade animals, the best way to do that, if it is a bloodline rather than a trait, is to understand how easily this can become diluted if you're not careful with how you pair things. And so it's frustrating now that my collection is primarily blade bloodline. I have blade in so much of my stuff that every time I post an animal or sell an animal, it's frustrating to have to explain the whole thing, at least being differently. I don't want to advertise something as a morph if I don't believe that it is. I'm comfortable selling something at a premium if it's nicer, which I believe that it is in that same regard. But I don't want someone to misinterpret what they're buying. And so it takes as much explaining as I just did, and most people don't even if they understand where I'm coming from and disagree, a lot of times they don't understand what I'm saying, because there's not a whole lot of bloodline understanding, selectively breeding more non-related to monogenic traits that I think that gets lost in ball pythons, which is tragic. There used to be, there's been these swells of it over the generations of ball python breeders where people have wanted to have, it's been very important to have the nicest version of single traits and combos. And then, new people come and old people go, and a lot of times it's just forgotten, and there's so many traits out there. A normal is a very rare thing these days. A lot of people don't consider the fact that bloodline has an impact on the outcome of the snakes that you're making, and that's fine. I mean, I guess I'll just keep doing my thing, but it's something that not only do not a lot of people appreciate, but a lot of people just don't understand that it's a possibility to selectively breed for nice looking examples of individual traits. In fact, I can't think of really any animals that I have that I bought as combinations. I bought my entire collection. I'm old now, but I bought my entire collection as base traits. And there were times too where, I had friends that were breeding things and they were getting all these cool things. And I'm here in the teens buying like a normal leopard ball python because I liked how reduced patterned it was. And, they're like, what are you doing, man? You don't need this stuff. Why are you building your collection out of $200 snakes? But it was important to me to start with nice stuff.
There's a whole lot to be said about not, instead of just ticking a box on the hets you want, the genes, the yes/no genes versus like refining something that you really like. Okay, this may or may not be a hot take, but there are some like crazy combos that it's cool and amazing that they've done that, but also it's not like if you just saw that from across the room, you'd be like, "Oh, that's." Oh, how do I say this? You know, like when someone has a baby and the baby.
You don't have to be nice. I don't think it's a hot take to say that some of them are ugly, even if they have a ton of cheese.
Yeah, like, you know, every baby that's born is precious, but you know, sometimes you have a ugly baby come out. You know,
right.
You love them, but
like
you know,
a GHI Mojave was amazing, and then some GHI Mojave combos people have made actually make it less appealing.
Yeah,
that's fair. Yeah,
add more stuff didn't make it better sometimes.
Yeah. Especially if you're not careful what stuff, even outside of selectively, you know, bloodline breeding and whatnot.
Well, with the blade problem and selective breeding, we don't have tests that will help you on that, but hopefully having an actual NoCo special test available now, hopefully you'll at least be able to talk less about what NoCo is.
Yeah, you saved half of my time in dealing with customers. I really do appreciate it. Now it's a matter of you know, I talked to Marshall about the. I let him know that it proved out to be a different genetic trait than special, and I pitched the idea of rebranding it, and he seemed a little bit apprehensive with the thought that maybe it would be a little bit confusing. I don't think. I think in the long run it makes more sense to give it a new name. Maybe it'll cause a little bit of confusion right in the beginning, but you know, like I said, I'm not worried about anyone that can't really wrap their head around that too much.
Well, let us know in the comments, all of you. I told Kyle before we started recording, my vote is just to call it NoCo. And you know, the full name,
I suppose, but if he I'd be thrilled if he could just call it NoCo. And it's not because the name special is stupid. It is stupid, but that's not why. So hopefully, I mean, it's up to him. I can live with it being called NoCo line special, but it would be really cool if it was just called NoCo or you know, any old name he decides.
But yeah, let us know in the comments. That'll be a suggestion and might affect the decision. But I that makes the most sense to me. Just call it NoCo, and when you write it out, I picture in Morphpedia and Morph Market saying NoCo, and then it says NoCo Special and talks about that, and the fact that it's very similar to Special, but the combos, especially the Super, the amount of contrast in the Super and pattern is very different from the big line. And to me, it seems like TCR and Baker are more similar to each other than either are to NoCo. So that's pretty cool to see.
I think so too. I mean, this is kind of how I felt, but it's a gene that I'm personally working with, and so you know, I'm hypercritical of being influenced by one's own interests, kind of, and so you know, I liked it, but I didn't think too much about it, and so it's nice to have concrete evidence. You know, it was one of the first things I thought of when I heard that you guys were doing this genetic testing thing. Was cool. Maybe I'll have an opportunity to prove out special. And then I saw, you know, special on your list and immediately messaged you guys. Let me send NoCo specials to see if they prove out, you know, as special or not. And,
yes. And we try to have, in most episodes, have a shed donor shout out. So this is an especially powerful shed donor shout out. We say thanks to Kyle Frost Reptiles, because that really is what made this possible, because you did the work. You message back and forth, and you know, sometimes there's so many things going on in our lives and your lives, and it's hard to connect and have a good conversation, be like, okay, let's put together a plan and make this happen. And thankfully it worked out, and we have a nice clean test, and it makes very good sense, especially because of what we know about the other blue-eyed Lucy mutations. It's right there and makes great sense, and definitely is not the exact same as special. It is the Baker line. It is definitely different. So
no, nice. Thank you guys. I mean, it only cost me a stamp, so you know, this is help me. Like I said, you cut down on my response time to customers. So, I appreciate all you guys do. I mean, I want to say thanks for making this kind of this technology, I guess, available equally to everybody. I remember when it first came out thinking like this could potentially be harmful if it were only made available to certain people. But
Yeah.
I mean, probably makes more sense from a business standpoint to make it widely available. But this has really been a lot of fun. It used to be that if I wanted to make a tri-stripe, puzzle, clown, genetic stripe, orange dream, leopard, yellow belly, I would have to, exercise and eat healthy to try and live long enough to make that happen. But now with this, I can do a podcast and eat a piece of cake and, still achieve my dreams in time. So this is great.
Eat trash and breed bonds.
That's right. That's me.
Heck yeah. So we stumbled onto this picture and Ben and I wanted to ask you about this insane looking animal. So you think she's a Partho? Yeah.
So and I have more questions than answers on this one too.
Yeah.
This was from a female leopard special orange dream that had a small clutch, and this was the sole survivor. Ooh. And I want to say it was, I forget what male, I bred a young male to it, but it was of a recessive trait. It nothing with NoCo. And so the fact that this popped up was totally unexpected. And so having, I'll start further back, having come from a boa constrictor background, Parthenogenesis is fairly common in breeding boa constrictors. In similar results, you get a smaller litter of snakes, and you get this anomaly that doesn't make sense given the male that was paired with the female. And often, these animals are generally born a little bit smaller. They're a little bit weaker. Sometimes they'll kind of just be a little bit soft in general. They can thrive to some extent. But they always seem to be either homozygous or wild type for any given mutation that the mother has.
Yep.
They typically, 99% of the time, end up being female. And having had experienced that several times, this is a very familiar experience. Is my first and only suspected parthenogenic ball python. But it was very in line with what I had experienced with boa constrictors in the past. And so looking at it, and I'm sorry, the mother of this one was an Inchy orange dream leopard special. And so looking at it, I saw the head pattern, I figured, and not knowing if the same holds true, if it's the same mechanism by which parthenogenesis occurs in ball pythons as it does in boa constrictors. If it is either homozygous or or wild type for any given trait. Looking at the head, I would assume that it is a super Inchy, and the pattern maybe a super leopard. Outside of that, I don't know if it caught orange dream. Kind of not important in this combo. Super special, obviously. It was a female, but very neat looking. It actually is, and especially in general, ball pythons carry their baggage better than boa constrictors do when it comes to having a ton of genes, especially if they're, deleterious in any sort of way. Not that any of these genes in this are, but I was surprised. It seemed to thrive very well. Kind of a novelty. I was at a time where I didn't have a whole lot of space. It's a really cool looking snake. I ended up selling this one to somebody that was super excited about it, and I should check up with them to see how she's doing, or if she ever went on to reproduce. It may not have been long enough ago to know that, but
if you do, reach out to them, give them our address. If they send a shed in, I'll run for Inchy and leopard, and if it's homozygous for both, then that's even more evidence they've got a part.
Yeah, but cool. That's unreal looking. Like that's so cool.
Yeah, I want to say I used like a tri-stripe male on the female, so it really made no sense that any of these genes would be present.
Yeah,
makes sense.
Cool. Gave me hope about the projects that I'm doing where I am intending on having all of these genes included.
Yeah. Yeah. All right. Let's see. I love this one. I love how the leopard and super are just so nice together with that.
Probably my favorite combo, aside from the, I don't know if you can call it Parthenogenesis, produced ball python your favorite morph. Maybe it is, but of the ones that I'm keeping and raising up and breeding, leopard's been my favorite combo.
Yeah.
Oh yeah.
Definitely, it's got a really cool kind of lava lamp pattern to it. It has the same head pattern as the regular leopard specials, and so it's weird that when in a super form you can actually retain kind of like what the indicators are in its heterozygous form, rather than it just kind of obliterating it like it would in, a lot of other super combos.
Yeah, and in this one, I can kind of see the speckle that you're talking about, that they tend to develop as they grow.
Not mites, I promise.
No, they're definitely scales. And also, I was thinking about it. I think the head stamp looks like, for me, it looks like a bicuspid, like viewed from the side a little bit.
Okay.
Yeah.
Pretty cool.
But yeah.
And she's kind of covering up her head, but and she's super special, and that's post ovulation and pre-lay. So it's a full-grown girl. Yeah, looking good.
Beautiful.
She's very, very full of eggs.
Yeah, and she's gravid again right now, and she's laid a big clutch of eggs this last year when the photo was taken, or the year before, and so the supers are strong. They thrive. They lay eggs. All that cool stuff. It's kind of nice,
Mm-hmm.
Even though I've had all these specials for all these years, it took me a while to actually get around to making supers. And then you always wonder in the back of your head, are they going to be strong? If I never had one. With all of this history of some genes not being super strong in their homozygous form, it's always nice just to experience that yourself. And so, So this project has never let me down.
That's awesome.
And when I saw this picture, I thought this was really interesting.
Yeah.
So these hatched out siblings to each other from, forget the exact pairing, but Orange Dream special to special, omitting the other genes. And so when they hatched out, my first thought was looking at them like, oh, one on the left is oranger. So
must be the
they're different. So this must be the Orange Dream one. And the other one must not be. And I sent it, but I'm not certain enough. And so I sent in sheds to you guys to confirm just for Orange Dream and also for your guys' tests for NoCo. Mm-hmm. And to my surprise, the one on the right proved out to be Orange Dream and the one on the left didn't. And
that's so wild.
When you look back, it's funny. When you look back on stuff like this, you could see, how you were maybe misled yourself. But the one on the left it's prettier, it's kind of oranger, but it is in line and consistent with what I expect and what I see with super NoCo line specials. And the one on the right, it's different. It kind of stands out more than what I would expect of a typical super NoCo line special. And then after a little bit of size and knowing the difference between so that's got a shed or two and a couple of meals in it, and it maintained and kind of built that contrast to it a little bit. So then it made a little bit more sense to me as it put on size.
Like if you go to the next slide, Kayla, and just kind of bounce back and forth. I mean, that amount of contrast the Orange Dream brings in, that's amazing. Yeah. I never would have guessed that.
Yeah, fresh out of the egg, like I said, I was mixed up between the two. So I'll have to I was able to look at it and be like, I can kind of see what I'll be able to use as visual indicators on this. And so the tail patterns kind of got the Orange Dream cleanliness to it and boldness to it. But it may be something that, especially with other genes involved, I'll just have to send you guys, $50 or whatever to make sure I know what I'm talking about.
It's 40 now. We dropped the price.
Yes.
Yeah.
All right.
Well, I'm thinking if you add leopard to the OD Super NoCo, I think that would be insane.
Absolutely. I think, the more you add to it, even all this little stuff I'm looking at, like, I'll be able to see clown in that easily. I'll be able to see puzzle in that easily. And so I do have a lot of NoCo het puzzles and NoCo het clowns and NoCo het this and that. So I'm really excited about the future of this.
For sure. Like I love how that I like an animal that maintains its pretty color, or or it matures and continues to look even better as it
Right.
grows. Like I think that's one thing I love about, my black rats. I always go back to those, but I especially love like in this OD Super NoCo, like that it continues to maintain that contrast.
Right.
That is gorgeous. So you
Very
got a
fun. A lot
very
of that stuff just washes out, but I think good bloodlines
Mhm.
and picking the right genes to add to it, I feel like it's pretty limitless. I tend to not take on genes where I don't see a long-term future in it. There's a ton of genes that I find super fascinating, but
Mhm.
the cool thing about ball pythons, there's enough genes that work well long-term that the projects that I'm in and the projects that I'm interested in are ones that I'll be able to do for the foreseeable future. It's just always something to do. There's nothing worse than kind of having a sense that you've done it all and there's nowhere to really go, which happens kind of quick with other species of reptiles.
Mhm.
There's just nothing compared to ball pythons when it comes to the just huge array of different genes. It's really no comparison. It's a lot of fun. You get to be picky within your species. And
Yes.
I only like 10% of the genes, and I still have plenty of genes that I like.
Yes, you do.
Awesome.
Well, thanks so much for coming on today.
Thanks for having me on. This has been fun.
Yeah.
Thanks for doing all the work.
Well, we love what you've done with NoCo line special, and I think a lot of people that see this, we maybe our thumbnail will be "Got NoCo." We're digging NoCo.
Yeah.
I think a lot of people. I think it's kind of been a hidden gem. It wasn't the first one. There's one that was similar to that went first, and so I think a lot of people just missed the boat, kind of like with Black Pastel versus Barnhart Black Pastel. Now we know that's a separate mutation, and we have a test for it. People can purchase something and know that it is that line, and it's just a little bit different, amps it a little different way. And I think that this is a very similar situation, and maybe even more extreme difference than with Barnhart and normal Black Pastel. This is really, really nice.
Well, I think it's cool, and it's nice to not be the only person that feels that way. So,
You've probably been feeling that way for a long time, huh?
The tumbleweed salesman from Family Guy. So.
Tumbleweed salesman. I forgot about that.
My time finally, but no, it's cool. It's a lot of fun. It's really exciting to be able to prove it out as something different, especially not having to do any of the work myself, and to a little bit vindicated my own obsessions over the years. This is cool. The future is fun in not all ways, but in this one way, it certainly is.
Definitely.
Awesome.
All right. Well, if you guys like this episode, be sure to like, subscribe, and comment. Let us know what you think. Let us know if you're smitten with the NoCo specials like I am, like Ben is. And certainly like Kyle is. So we're really, really grateful to all of you guys for tuning in and for for your support, letting us do what we do. So you guys got anything before we call it a day?
No, that's it. It's the most I've talked about snakes in years.
Awesome. That's a good feeling.
We're very thankful you did it on here.
Yes.
Well, thanks for having me.
All right. Well, thank you guys, and we'll see you guys next week. Outro in 3, 2, 1.